channel. Today we are going to discuss about genetic engineering. What is genetic engineering? Before we discuss about genetic engineering, first we have to know about genetics. What is genetics? Study of genes. Study of genes are known as genetics. Understand? Is this clear? What are genes? Genes are made up of DNA. Genes are made up of DNA. It will carry the information. It will carry the information from one generation to another generation. Okay. Now let us discuss about genetic engineering. What is genetic engineering? Some modification takes place in genetics. That is called genetic engineering. Some modification takes place in genes. That is called genetic engineering. Genetics. Natural process takes place in genetics. Genetic engineering, artificial process takes place in genetic engineering. We can alter the genes, we can modify the genes because of some reasons. Okay, genetic engineering are also known as recombinant DNA technology. Recombinant DNA technology are also known as gene cloning. Gene cloning. What is genetic engineering? Gene modification. Gene modification takes place in genetic engineering. What is rDNA technology? Transfer of gene. Transfer of gene from one organism to another organism takes place in rDNA technology. What is gene cloning? It can produce multiple identical copies of gene. Multiple identical copies of gene. These are the collective term for genetic engineering. What will happen here? Gene modification. What will happen here? Gene modification. Understand? Is this clear? In genetic engineering, modification of gene takes place here. We can transfer the gene from one organism to another organism by using these instruments. These are all instruments used for transfer of DNA from one organism to another organism. What are the instruments? Electroporation, gene gun, liposome and chemical mediated transfer and microinjection. These are all the instruments help for transfer of DNA from one organism to another organism. These are all the instruments used for transfer of DNA. Understand? Is this clear? Okay, let us move on to next one. rDNA technology. Now let us discuss about what are the steps involved in rDNA technology. What is rDNA technology? Recombinant DNA technology. Recombinant DNA technology. What is the meaning of recombinant? Rejoining. Rejoining of DNA. Okay, rejoining of DNA. This recombination process occur naturally. Recombination process occur naturally. We can see the natural recombination process in meiosis. We can see the natural recombination process in meiosis. But we can do the recombination process artificially by using some modern technology. Okay. In this technology, we can transfer the DNA from one organism to another organism. Can you understand? Is this clear? In DNA technology, we can transfer the DNA from one organism to another organism. Okay, now let us discuss about the steps involved in DNA technology. Now let us discuss about what are the steps involved in DNA technology. See here, this is the bacterium cell. This is the plasmid vector. Plasmid vector. What is vector? Vector used as the carrier. Okay, it has the ability to carry the DNA. Understand? Vector has the ability to carry the DNA molecule. We have to isolate the plasmid from bacterium cell. Okay, we have to isolate the plasmid from bacterium cell. See, this is the donor DNA. Okay, we have to cut the specific DNA. See here, we have to cut the specific DNA by using restriction endonuclease. Restriction endonuclease. This enzyme used for cut the DNA molecule. Understand? Is this clear? Restriction endonuclease used for cut the DNA. If we want to cut the things, we used knife, scissor, etc. like that. If we want to cut the DNA, we should use restriction endonuclease enzyme. This enzyme used for cut the DNA molecule. By using this enzyme, we can cut the DNA. Okay, now we isolated the plasmid. We cut the DNA. Okay, now we are going to join the plasmid with DNA. We have to join the plasmid with DNA. This joining process needs ligase enzyme. Ligase enzyme. This ligase enzyme used for join the DNA. Join the DNA with plasmid. Ligase enzyme used for joining process. Restriction endonuclease used for cutting process. Understand? Is this clear? Now we are join the plasmid vector with DNA. Now we got rDNA. This is the rDNA or recombinant DNA. Then we have to insert the rDNA into the host bacterial cell. Okay, we inserted the rDNA into the host bacterial cell. After that, plasmid replicated here. Plasmid vector have the ability to replicate inside the host cell. Okay, gene cloning takes place here. Okay, understand? Is this clear? This is the rDNA technology. See here, by using this rDNA technology, we can produce many medicinal products. Medicinal products. For example, we can produce medicine for asthma, anemia, diabetes, ulcer, hepatitis, kidney disorder, immunity disorder, etc. Okay, by using this rDNA technology, we can produce many medicinal products. Okay, I'll give an example. Technology, we can take diabetes. Okay, we can produce insulin by using this rDNA technology. Okay, I'll give an example. See here, we consider this is the insulin coding gene. This is the bacterial plasmid. We have to take the insulin coding gene, then we have to isolate the plasmid. Okay, then we have to join the plasmid vector with insulin coding gene. Okay, then we have to insert the rDNA into the bacterial host cell. Inside the host cell, this insulin coding gene will replicate. Finally, we collect the insulin gene. Okay, like that, rDNA technology helps to produce medicinal products. Understand? Is this clear? Thank you. We will see in next video.